welcome to the Modulus Research YouTube channel. Thanks for watching. Today we're going to be talking about how to write a research proposal. So there are three essential components of writing a research proposal. In less than 5 pages, you can obviously modulate that to make it longer. But before you get into actually writing your proposal, you want to start with finding sources, because that's how you're going to find the content that you need to write your introduction. So where can you find sources? You can start by looking at databases like NCBI, PubMed, NIH, Google Scholar. We can put a whole list here for you guys to look at, but these are good places to start when you're looking for content for your proposal. The first part of your proposal is going to be the introduction, and that's where you want to conduct a literature review of the topic that addresses the following. First, what is your research about? Give us some context. Summarize in 3 to 5 sentences. So we're going to take you through this example involving Wnt signaling. So the first thing you want to tell people about is, well, what are Wnts? Wnts are these short-range signaling molecules, proteins that are important during cell differentiation, morphogenesis, renewal, propagation, etc. Next, you want to take them through the journey of Wnts in the cell. Why are they secreted? What's their role within the cell? And you want to end with some sort of tying back to why do we care? And that kind of leads me into the second point of you want to tell your audience, why do we care about this topic? Summarize this in 2 to 3 sentences. So in the case of Wnt signaling, if you dysregulate this pathway, that can cause developmental issues and oncogenesis, particularly in colorectal cancer. It can also lead to developmental disorders like Zaki syndrome in mutations in Modulus. So these are both examples of the big picture of why we want to read about the experiments you're proposing. The third point is, what in the experimental method would you be using to study this topic? So you want to make this one sentence at the end of your introduction to summarize that. So, for example, this study proposes the structural elucidation of apo-Wnt to understand how one dedicated carrier can accommodate 19 different proteins and provide insight into how anti-cancer therapeutics can fit into the binding pocket. This sentence combines both, the specifics of the project that you're studying as well as why we care about it, and that's why it's important to state at the end. So basically, be succinct, be economical with your words, don't give away too much information, and give us the elevator pitch in one sentence. The second section is writing your aims. So you want to propose a list of maybe 3 to 5 aims. Usually 3 is a good number for a short proposal, and you can subdivide them as needed. So step 1 is title your aim with your intended group of experiments, and it should be broad enough that you can elaborate in detail with the subsections that follow. So for example, one title could be Aim 1, Preliminary Screening of Wnt to Optimize Yield in Small Scale. Second step would be to aim to be specific as possible with your procedures. So you want to get into the details of viral titrations, incubation times, the reagents you're using, the cell lines you're using. These are all really important because they show your reader that you're very well informed about the subject that you're talking about, and you've thought in detail about how you could execute this experiment if you were going to. Don't stress about actually doing the procedure. This is simply a proposal. It's not about actually executing it, but you want to show your reader that you have some idea of how to actualize this goal if you were going to. Final step in writing your proposal is to write about your future studies, and this is arguably the most important part of your research proposal because you want to conclude with how this experimental information will be used going forward and what future work can be done in this field. So the very first step would be consider the data or results that you may obtain from your proposed experiment. How would you interpret them to lead to your desired next steps? And of course, you aren't actually doing this experiment, but think about what you might do and how that expectation can inform your future projects. Next, you want to think about the logical next steps for your project. What experiment could be done contingent on the experiments that you've obtained? So for example, if we're talking about this cryo-EM structure of Wnt, you might be considering, well, how can I validate this structure? How can I make sure that this is a structure that could potentially be obtained in a physiological environment? So you might say, okay, I will use all these validation tests to make sure that this is the structure that is actually present in biology. The next step is we want to propose some more complex experiments that are a little bit more out of the box, innovative, that shows your reader that you're thinking past what could be a logical next step in the field that might be the convention of the field. You want to think about something that might not be as standardized. So, for example, in the case of obtaining this cryo-EM structure, you might think, oh, let me try and cross-link two helices and compare, if the ligand is accessible in this cross-linked structure versus in another structure, and see how those two models compare. The third step you want to take is put a disclaimer about your research. Are there caveats? Are there limitations? And how can you still glean some relevant insight from your experiments? Because of course no project is perfect, but you still want to make that argument that your research is still worthwhile in pursuing. So for example, if you wanted to talk about cryo-EM and say, okay, well, why should we actually trust the models that we're putting out there? How are they physiologically relevant because we're obtaining them in sort of this like artificial environment? You can say, of course, we're never going to fully know if a protein adopts this structure in its native environment, but we can take steps to further corroborate the confidence that we have in this model. So thank you guys so much for watching. Be sure to drop your questions in the comments below, like, subscribe, and we'll see you guys next time.